Now we're beginning Chapter 2 of Emerging Viruses, AIDS and Ebola, Nature, Accident, or Intentional. Who plays in the big leagues? Jackie, my wife and co-investigator, had been instrumental in helping me research the Florida dental AIDS tragedy for deadly innocence. The loving mother of our now two children, Jackie began her working career as a dental assistant for the Saskatchewan dental plan in Canada. We met in Cancun, Mexico, waiting in line at Carlos and Charlie's bar and grill. At the time, she was looking for a job and I needed an assistant. The rest is history. Besides her big blue eyes, long silky auburn hair, slight build, and innocent appearance, what attracted me most about my future wife was her survival instinct. She had spent almost 2 months touring the back roads of Mexico virtually unchaperoned. The girl's a survivor, I respectfully considered. Over the years, I found this trait increasingly comforting, particularly while comforting the many frightening realities we encountered during our research. The who does what? The only thing I know about the World Health Organization, I said to Jackie after learning of Strecker's theory, is that it's a prestigious, internationally supported organization that develops health and vaccination programs for developing countries. It suddenly seemed odd to me that over the course of my training, more than 4 years of college, 3 years of dental school, 10 years of postdoctoral research and teaching, and 16 years of clinical dental practice, I had learned very little about the WHO. I don't even know what's involved in becoming a WHO member. I admitted the name sure imparts an air of scientific aristocracy. Eventually, as the novelty of Strecker's theory wore off and further attempts at contacting Strecker by phone failed, I decided to venture into the dungeons of Harvard's Countway Medical Library to prove the null hypothesis that nothing was true about Strecker's memorandum. What I unearthed, however, in the back issue of the WHO Chronicle was engaging. Dozens of WHO Chronicle articles that I photocopied and brought home revealed that by 1968 the WHO had solely in control of the world's experimental biologicals for almost 2 decades. WHO has exerted a powerful influence on the quality control of biological substances since its very inception in 1948. Since 1952, the WHO interest in the establishment of international requirements for such biological products began. Various possible measures have been examined for attempting to achieve a greater degree of uniformity in the quality, safety, and potency of vaccines, antisera, etc., for the control of substances of particular interest to WHO in relation to its mass immunization and mass prophylaxis schemes in developing countries. The main purpose served by these international standards, reference preparations, and reference reagents is to provide a means of ensuring worldwide uniformity in expressing the potency of preparations used in the prophylaxis, therapy, or diagnosis of human and animal disease. The coordinating body for all this work, I learned, was the WHO Secretariat. The Geneva-based organization maintained several full-time officers and part-time consultants who worked in collaboration with several other laboratories in other countries. The laboratories most deeply involved are the WHO International Laboratories for Biological Standards within the Departments of Biological Standards of the Staten Serum Institute, Copenhagen, the National Institute for Medical Research, London, and the Central Veterinary Laboratory, Weybridge, England. Between them, these laboratories undertake the detailed work of organizing international collaborative essays and holding and distributing the international biological standards and many of the international biological reference preparations and international biological reference reagents. The initiative for setting up standards and reference preparations usually comes from the WHO Expert Committee on Biological Standardization, which is convened annually in Geneva. It comprises recognized experts in the field who serve annually in Geneva. It comprises recognized experts in the field who serve without remuneration in their personal capacity and not as representatives of governments or other bodies, together with members of the WHO Secretariat. This expert committee also establishes the international standards and reference preparations on the basis of the results of the international collaborative essays. For pharmaceuticals generally, still including some biologicals, and drawing up of standards is in the hands of the expert committee on specifications for pharmaceutical preparations in collaboration with the WHO Secretariat and with the help of the expert advisory panel on the international pharmacopoeia and pharmaceutical preparations. Needless to say, close liaison is needed between the Secretariat, the Expert Committee on Biological Standardization, the Expert Committee on Specifications for Pharmaceutical Preparations, and various other expert committees on, for example, antibiotics, tuberculosis, yellow fever, and cholera. Another article discussed the WHO's national control activities, which provided advice and encouragement when countries became conscious of the need for controlling biologicals. WHO helped them establish and develop their national control laboratories. It was quickly apparent that the WHO set the standards for the development, manufacture, distribution, and administration of essentially all pharmaceuticals used throughout the world. As seen in Figure 2.2, they were also intimately involved in determining which drugs should be made or remain illegal. Besides assembling teams of scientists to develop, test, and standardize new and ancient drugs, the WHO applied similar administrative leadership to develop plans for attacking all the woes of humanity: polio, yellow fever, cholera, smallpox, whooping cough, diphtheria, tinnitus, measles, anthrax, typhoid, tuberculosis, influenza, and even the common cold were all targeted. The WHO's approach to controlling communicable disease was spelled out by their Assistant Director General, Dr. A.M. Payne. Mass campaigns against certain communicable diseases require an initial attack sustained interruptedly over a relatively large area within a short period of time. In smallpox, for example, the buildup of new susceptibles in the absence of routine vaccination creates an explosive situation, resulting in the familiar pattern of epidemics of smallpox followed by the epidemics of vaccination. WHO's Developing Viral Network. Applauding WHO's support for pioneering work in viral research, Dr. D.A. Tyrell reported on common cold rhinovirus provided valuable insights in the growing field of virology. In the early 1960s, WHO designated Tyrell's research unit in the United Kingdom and the National Institute of Health, NIH, in Bethesda, Maryland, as two international reference centers in order to promote their respiratory virus study. From here, newly developed techniques for virus cultivation, Tyrell wrote, were widely applied. Hundreds of strains of rhinoviruses have been isolated and shown to be antigenically distinct from at least some other strains. They have been reported in the scientific literature under a confusing variety of designations, and it was accordingly decided at a meeting of the directors of the WHO Virus Reference Centers to undertake collaborative study in which sera and strains were distributed to a number of laboratories so that cross-neutralization tests could be performed of a well-characterized and apparently new strains. This work was supported by the Vaccine Development Board, emphasis added, and coordinated by the WHO International Reference Centers. Work on these viruses, Tyrell continued, demanded a supply of cells that were sensitive to such organisms. It required considerable work to find such cells. Often cell lines would change their sensitivity after prolonged cultivation. The reference centers thus maintained stocks of cells stored in liquid nitrogen, which they distributed to labs conducting viral research throughout the world. Some viruses that failed to grow in the usual tissue cultures, Tyrell revealed, were propagated in cultures of the human trachea and nose, that is, in the organs and tissues in which they multiply in nature. These viruses, some new rhinoviruses and other new types, never before detected in man, were disseminated through the WHO network of virus reference centers. So let me get this straight, Jackie said. World-renowned scientists developed WHO policies and practices, studied and distributed viruses with financial support from groups like the U.S. Vaccine Development Board. Was the board, like the WHO, connected to any pharmaceutical companies? I'm not sure, I replied, but most likely there was obviously lots of money to be made with vaccines, and only a few companies made them. Which ones? Well, Merck, Sharp, Dome, MSD is one of the largest, and they did fund the hepatitis B vaccine research. Streckert alleged spread HIV to homosexuals in America. Another report 4 months later showed Israeli scientists were supported by the WHO to study the genetic determinants of the human immune response. A few others stated that the WHO was funding several programs designed to evaluate the specific diseases, vulnerabilities of minority groups, from American Indians to African natives, through the collection and analysis of gene pools and blood supplies. That's just what the Nazis did, Jackie recalled. Here's a couple more articles noting the WHO and the U.S. Vaccine Development Board also funded large-scale human trials of newly developed vaccines made from both bacteria and viruses. Let me see. I passed the reports over to my co-investigator. Just as Streckert reported, Jackie said after reading the articles carefully, yeah, I hate to say it, but maybe there's something to his theory. Their smallpox eradication program used vaccines made from antisera made largely in the United States and given for free to African countries, including Kenya, Ethiopia, Guinea, the Democratic Republic of the Congo, and Rwanda. The Democratic Republic of the Congo, which eventually became Zaire, they said would have a sufficient production capacity to supply the needs of all African countries south of the Sahara. That's interesting and very notable, Jackie retorted, somewhat cynical. Zaire, the center of the African AIDS belt, supplying neighboring countries with the technology and expertise they needed to become healthier and more self-sufficient, is great. I only wonder who paid for it and why. I just read that their Vaccine Development Committee endorsed a 1970 African campaign budget of $14 million, I answered. That was a lot of money for those days. About how much in present dollars? I asked my more mathematically gifted partner. Say about 5 times that, around $70 million. Much of it apparently came from the United States and other world governments interested in Africa, and periodic infusions of more cash for revaccination campaigns were needed and supplied. The Louisiana Laboratories. In 1964, shortly after President Kennedy's assassination, the WHO created International Reference Center for Immunoglobulins at the University of Lausanne, Switzerland. 3 years later, the WHO Regional Reference Center of for Immunology Research and Training, was designated at the same site. Its director, Dr. Rowe, reported that the center was established to broaden the WHO's range of activities insofar as the study of antibodies and immunoglobulins, the naturally produced proteins that defend the body against attack by toxins and germs. Rao noted the WHO's special interest in cell-mediated immunity, that is, the cells that reorganize antigen, foreign proteins associated with germs and toxic substances, secrete antibody, and are themselves able to attack foreign cells. Primary defense cells, called lymphoid cells, Rao noted, were under intensive investigation to determine how they initiated and maintained the immune system, paramount in determining the pathogenic effects of infectious agents ranging from viruses to parasites. Apparently their experiments went well, I remarked. In December 1969, the WHO issued its second five-year research report on viral experiments it had funded or conducted since 1959. The report stated, in the 3 years, 1964 through 1968, the principal advances in virology were in knowledge of the fundamental structure of viruses and cells and of their interrelationships and interactions. A much greater understanding was gained of the natural behavior of viruses as infectious agents, of pathogenesis of viruses, disease, and of the means of controlling many of the common virus diseases, generally by improving existing vaccines or by developing new ones. Through direct proof of a causal relationship between viruses and human cancer still escapes the numerous investigators working on this subject, the quest continues to be energetically pursued. The hypothesis that at least some malignant neoplastic diseases, such as leukemia, are associated with virus infection is perhaps even more strongly expressed now than in the past. The article went on to state that Russian and American researchers were privy to the same vaccines. Viral samples and information about how the human immune system could be bolstered or destroyed by old and newly developed germs, including those produced from monkey viruses. All this during the Cold War, Jackie noted. Green monkeys, slow viruses, and $10 million. Strecker's materials said that the D.O.D. provided one contract in 1970 for $10 million for the development of a synthetic biological agent with no natural immunity. The WHO's reference center got that? Jackie asked. It had to have been one in the U.S., for sure, but where? There were only two possibilities, I said, Atlanta, Georgia, and Bethesda, Maryland. The Atlanta lab was run by the CDC's predecessor, the National Communicable Disease Center, NCDC. The Bethesda lab was run by the NIH. The later was cited in the WHO Chronicle as one of the initial two The Bethesda lab was run by the NIH. The latter was cited in the WHO Chronicle as one of the initial two international virus reference centers. Yet it was reported to be inadequately equipped to handle dangerous smallpox viruses. These were allegedly handled in Atlanta. If that's the case, it's not likely they would have handled deadly viruses like HIV either, Jackie reasoned. Not necessarily, I responded. The smallpox virus and the DoD requisition may have posed different risks. Shortly after our conversation, an article by Charles Siebert in the New York Times Magazine clarified the biological safety level (BSL) risk rating system used by the CDC and the NIH. In its hierarchy of precaution taken against biological threats at the CDC, BSL 1 and 2 are the lowest level of safety. Work is done there only with non- or moderate-risk organisms. Viruses that cause colds, for example, or bacteria that cause diarrhea, at BSL-3, known as the hot zone for the blue suit lab workers, visit the highly transmittable viruses or with those viruses or bacteria for which there is no known cure. There are only 2 BSL-4 labs in the country, one in the United States Army Medical Research Institute for Infectious Disease, USAAMRIID, in Fort Detrick, Maryland, and one in Atlanta. It's considerably more now. We've got more than 10. Our road atlas showed us Frederick was very close to Bethesda. I picked up the telephone to learn more. An administrator at the NCI, National Cancer Institute, Tumor Cell Biology Lab in Bethesda, confirmed Siebert's report. Additionally, the woman told me, the AIDS virus is considered a BSL-3 hazard. It's being studied in Bethesda as well as numerous labs across the nation. We also learned that once developed, the most dangerous viruses planned for use as biological weapons were shipped to the Pine Bluff arsenal for storage. Among the tens of thousands of viral strains cultured, developed, and transported for study by WHO reference centers, we learned that 2 received special attention and an inordinate share of research dollars. Monkey viruses, including the simian pox virus and the slow viruses, particularly Bisna and Scrapie. We read these reports carefully since Strecker noted the AIDS virus bears the greatest likeness to the human bovine cow lymphotrophic lymph cells targeting and cancer-causing virus combined with sheep Visna virus. Monkeypox was of great interest to researchers. The WHO Chronicle said, for 2 reasons. First, the monkeypox virus was found closely related to the variola vaccinia virus group, which causes and immunizes against human smallpox. Second, the monkey is man's closest relative in the animal kingdom, and experimental results using monkeys were expected to provide the best indication of what might occur in humans exposed to the same elements. Alternatively, slow viruses were of the greatest interest to WHO, CDC, NIH, and NCI scientists between 1968 and 1974. The reasons for this were not as obvious. The WHO Chronicle reported recent interest in the slow viruses, in particular those causing chronic degenerative diseases of the nervous system, the China chronic infectious neuropathic agents, viruses, has come from painstaking work with Visna and Scrapie degenerative diseases of the central nervous system of sheep, and Kuru, a degenerative disease of the central nervous system of man restricted to the 4 people of New Guinea and their immediate neighbors. Why so much interest in 2 sheep viruses that cause nerve disorders and don't infect humans, Jackie said. I'm not sure. What about Kuru? Who are the 4 people of New Guinea? What makes them so important that viral centers around the world took up their cause? Well, let's look it up. I walked over to the library and pulled out a copy of Stedman's Medical Dictionary. Kuru, it says, is a highly localized, fatal disease found in New Guinea resembling paralysis agitans, a nervous disorder with frequent bouts of shaking, found among certain cannibalistic people who ingest raw brain of recently deceased victims of the disease, also called a laughing sickness. When in history has helping cannibals been a world priority, I wondered. Never, Jackie responded. The notion seems utterly harebrained. Oh, that was awful. Sorry, I couldn't help myself. We read on. China viruses are distinguished by languishing character of the infection process they initiate. The incubation period in the host may be months or years, and the disease itself may progress laggardly towards an irreversible deterioration of the victim. Cells infected with slow viruses are, in general, neither prepared nor stimulated to proliferate. Their functions are impaired by the nature of the dysfunction. I gotta read that over. Their functions are impaired, but the nature of the dysfunction has not yet been clarified. It's remarkable how closely this matches several of the most prominent features of AIDS, I said, and there's more. The resistance of the scrapie agent to heat, ether, formalin, and other enzymatic and chemical agents, as well as its very small particle size, poses the question whether it is a conventional virus, an incomplete virus, or some other agent. The findings of different research groups are at variance and in several instances are totally inexplicable within our present concept of infectious agents. That reads just like the D.O.D. order for a new infective microorganism that couldn't be defeated against, I remarked. The article went on to state that additional experiments had been conducted in order to prompt the human immune response by the injection of double-stranded RNA. HIV is a single-stranded RNA slow virus, I explained, and gene cutting and splicing techniques were well developed at that time. Could they have cut double-stranded RNA to make single-stranded RNA? I'm not sure. But what I don't understand is, here, the WHO Chronicles stated the primary objective of their viral research program was to acquire a thorough knowledge of the virus diseases so that the prophylactic and other public health measures can be introduced as soon as possible. What's the matter with that? Look at what they were studying to accomplish it. 2 rare diseases that only affect sheep and 1 totally remote virus that makes brain eaters laugh themselves to death. Do you think they might have been looking at these things for use as biological weapons? Jackie asked, and then added, think about it. Scrapie, a totally unconventional germ that they're not even sure what it is. You can't kill it with heat or chemicals, and there are still no tissue culture systems or antibody systems by which enemy defenses could be prepared. Added variance, and totally inexplicable with the current knowledge at the time, I added, the enemy would not only be surprised but baffled and helpless. We reflected again on the DoD document that detailed their desire to acquire a new infective microorganism which could differ in certain important aspects from any known disease-causing organisms. Most important of these is that it might be a refractory to immunological and therapeutic processes upon which we depend to maintain our relative freedom from infectious disease. It is a highly controversial issue, and there are many who believe such research should not be undertaken, lest it lead to yet another method of massive killing of large populations. The following week, we learned that despite heavy opposition by the public and House representatives, the United States Congress gave the Army $23.2 million for biological warfare research. What year was this? Oh, yeah, before Nixon. About half of that, at least $10 million of taxpayer money, went directly toward funding the manufacture of immunosuppressive agents, allegedly for defense. In essence, this one 1970 DoD biological weapons appropriation cost more than half of all the money the WHO spent in Africa that year for all of their healthcare and vaccination programs. Jackie calculated. Figure 2.1, requirements for biological substances. 1958, general requirements for manufacturing, establishing, and control laboratories, revised in 1965. 1958, poliomyelitis vaccine, inactivated, revised in 1965. Another 1958, cholera vaccine, revised in 1968. Another 1958, smallpox vaccine, revised in 1965. 1959, General Requirements for Sterility of Biological Substances. 1961, Poliomyelitis Vaccine, oral, revised in 1965. 1963, pertussis vaccine. 1963, Procrane benzyl penicillin in oil with aluminum monosterate, revised in 1965. 1963, diphtheria toxoid and tetanus toxoid. 1965, dried BCG vaccine. 1965, measles vaccine, live and measles vaccine inactivated. 1966, anthrax spore vaccine, live for veterinary use. 1966, human immunoglobulin. 1966, typhoid vaccine. 1967, tuberculins. 1967, inactivated influenza vaccine. 1969, immune sera of animal origin, to be published. Source: Matthews, A.G., WHO's Influence on the Control of Biologicals. WHO Chronicle, 1969, 23, colon 1, colon 3 through 15. Figure 2. The WHO's influence on the control of biologicals involving the development of the international standards regulating pharmaceuticals. WHO's influence on the control of biologicals. It seems to be a most appropriate time to review the work of WHO in relation to the quality of biological reports. In 1968, the organization completed its 20th year of existence. It is during its second decade that WHO has exerted a particularly direct influence in this field by virtue of establishment of a series of requirements for biological substances, international biological standards. However, in a somewhat less direct fashion, WHO has exerted a powerful influence on the quality control of biological substances since its very inception in 1948. The work of setting up and distributing international biological standards was not started by WHO, but was taken over already in an advanced stage of development from the Health Committee of the League of Nations. Indeed, for the first few international standards for biological substances were established by a national body, the Statens Serum Institute, Copenhagen, a few years before the creation of the Health Committee. The very first such standard, the International Standard for Diphtheria Antitoxin, which consists of a dried hyperimmune horse serum, was established in 1922 and it is still in use today. It says much for the forethought and wise choice of the early authorities, as well as for the stability of at least some biological products, that a single preparation has served world requirements for a period of 46 years. The supply of this particular standard is expected to last for at least another 46 years. For this small start in 1922 and up until 1948, when WHO was established, the number of international standards distributed by the League of Nations grew to 32. In the categories enumerated in Table 2, the total number of international biological standards issued by WHO is now 79, and in addition there are 56 international biological reference preparations. Also, in recent years, 96 international biological reference reagents have been established by WHO. Generally, these are intended as reference materials for substances used in the diagnosis of disease and in the identification of microorganisms. Many leptospiral typing antisera are included among these reagents, and a recently established set of viral typing antisera is being rapidly expanded. Table 2 gives a classification of the current and international preparations with comparative figures for 1948. In general, the main purpose served by these international standards, reference preparations and reference reagents, is to provide a means of ensuring worldwide uniformity in expressing the potency of preparations used in the prophylaxis, therapy, or diagnosis of human and animal disease. Most of the substances of which these international standards, etc., have been established could not, at least at the time of their establishment, be characterized fully by chemical and physical means. The activity of an ill-characterized substance may be measured by biological assay, and the results may be best expressed as a ratio for its activity to the activity of a closely similar physical specimen, designated the international standard, in many cases the definition of international. And this says, one of the numerous WHO Chronicle reports obtained from Harvard's Francis Countway Medical Library during an initial investigation into the origin of AIDS. Source: Matthew A.G., WHO's Influence on the Control of Biologics, WHO Chronicle 1969, 23:1:3-15. That's it. We'll begin Chapter 3 tomorrow.